I think we can continue to this webinar. As Miron said, my name is Dimitris Kardassis, and I'm professor of biochemistry at the University of Crete Medical School and affiliated member of the Institute of Molecular Biology and Biotechnology in Heraklion, Greece. Thank you very much for attending this this webinar, despite the fact that it's late in the afternoon for some of us. So in these first webinars, this is the second webinar, Paolo Magnis was the first one, we'll try to cover the basics in the pathophysiology of atherosclerosis. So in my webinar, we'll talk about lipoproteins and how they contribute to the pathogenesis of atherosclerosis. So the topics that we will cover today is brief introduction to atherosclerosis and cardiovascular diseases, risk factors for atherosclerosis, introduction to lipoproteins, classes, structure and composition, methods of separation, an overview of lipoprotein metabolism, the VLDL, we'll focus on the VLDL, LDL pathway and genes causing familial hypercholesterolemia. We'll talk about mechanism of action of statins and another recent drug called bempedoic acid. We'll talk about PCSK9, which is a new player in atherosclerosis and is a target of new potent drugs lowering LDL cholesterol. We'll talk about HDL and reverse cholesterol transport and genetic syndromes of low HDL levels, the atheroprotective functions of HDL also. And finally, we'll close with new therapies based on HDL functionality. So we'll start with some key facts from the World Health Organization. About 2 years ago, cardiovascular diseases, which are essentially ischemic heart disease and stroke, are the leading causes of death globally. An estimated 17.9 million people died from cardiovascular diseases in 1 year only, 2019, representing 32% of all global deaths. And of these deaths, 85% were due to heart attack and stroke. So this is summarized in this slide, which shows the leading causes of death globally. In 2 years, with a 19-year distance between them, 2000 and 2019, and you can see that ischemic heart disease and stroke take the first two positions, top positions in this list. And essentially, it was the same in 2000. So despite the fact that there are some recent progresses in drug development for cardiovascular disease, we see that after 20 years, still many people die from cardiovascular disease, and these are the top cause of death globally. The main cause of cardiovascular disease is atherosclerosis. I will say a few words because Paolo Magni in his webinar covered the topic very well. So just to put everything into perspective, atherosclerosis is the formation of atheromatous plaques on the arterial wall, such as the coronary, the carotid, the peripheral arteries, and the progression. And actually, atherosclerosis leads to progression of cardiovascular events, such as myocardial infarction and stroke. And this is a cartoon showing an arterial stenosis due to atherosclerosis. We see a very simple schematic representation of atherosclerotic plaque containing lipids and connective tissue, and we see that the diameter of the lumen has been decreased, thus impairing the blood flow. And atherosclerosis involves many stages, from foam cells to fatty streaks, intermediate lesions to atheroma to fibrous plaque, and in some cases of unstable plaques, complicated lesion rupture. Starts from first decade and continues to late in life. And the primary dysfunction here is the endothelial dysfunction. And this is very important for our discussion. And you can see here that during the first 3 decades, the growth of the plaque is mainly due to lipid accumulation. And this lipid accumulation that we see here in yellow is due to lipoproteins and primarily due to LDL, oxidized LDL. So very briefly, the timeline of atherosclerosis is endothelial injury, activation of endothelial cells that start expressing adhesion and monocyte chemoattractant proteins, migration of monocytes and lipoproteins. I have highlighted in blue the lipoproteins that we'll talk about today to the subendothelial space. oxidation of LDL by oxidizing enzymes in the intima, and clearance by macrophages via scavenger receptors, formation of foam cells, recruitment and proliferation of smooth muscle cells, which can also absorb oxidized LDL and become foam cells, synthesis of connective tissue from smooth muscle cells and formation of fibrous cap, and plaque rupture thrombosis. So here you see some highlights that show the involvement of lipoproteins to the pathogenesis of atherosclerosis. We'll talk about this in detail. So which are the traditional risk factors for cardiovascular disease? There are two main classes of risk factors, the non-modifiable and the modifiable. The non-modifiable include sex, male are more predisposed, age, more than 50 years, increases the risk, and family history, mutations in genes. You cannot do anything about it. However, you can modify the other risk factors, that is lipidemia. As this lipidemia, we define high levels of total LDL cholesterol or Lp(a), low levels of HDL cholesterol, and high levels of triglycerides. We also have diabetes, hypertension, obesity, smoking, among others. So today we'll talk about this important risk factor for atherosclerosis, which is dyslipidemia. We'll talk about lipoproteins and their metabolism and their pathways and their genes, and what drugs that are available today to modify these risk factors. So all this data about the risk factors for cardiovascular disease come from large epidemiological studies, such as the Framingham Heart Study, shown in this slide, which established, for instance, that low HDL cholesterol and high LDL cholesterol are two independent risk factors for atherosclerosis. Or in other words, low HDL cholesterol predicts coronary heart disease risk independent of LDL cholesterol. So the lower the HDL cholesterol levels, the higher the risk, which is shown on the vertical axis, in all LDL cholesterol concentrations. Now, these lipidemias are caused by disturbances in lipoprotein metabolism. And we'll continue in this slide to a brief introduction on lipoproteins. So lipoproteins are macromolecular complexes of lipids and proteins that are synthesized mainly by the liver and the intestine, and their main role is to transport free cholesterol, cholesterol esters, triglycerides, phospholipids, and other lipids derived from exogenous or endogenous sources in the circulation. The plasma lipoproteins are classified according to their density, and they can be separated based on their density, for instance, in ultracentrifugation. The protein constituents of lipoproteins are called apolipoproteins, and they form a family of proteins with similar structural characteristics, amphipathic alpha helices, but different functions. And lipoproteins can bind to different plasma enzymes, such as lipases, esterases, lipid transfer proteins, and can be remodeled. They can change shape and composition. So this is a structure of a typical lipoprotein particle. On the surface, we have the polar molecules, such as the phospholipids, free cholesterol, which has a hydroxyl group which is polar, and the proteins, which consist of amphipathic alpha helices, and the hydrophilic helices, they are from the outside, and from the inside we have the hydrophobic part of the helices. And in the core, all these lipoprotein particles contain the hydrophobic molecules, which are primarily triglycerides, shown here, and cholesterol esters. Cholesterol esters are derived from cholesterol when this hydroxyl group is esterified with a fatty acid by enzymes called esterases, and one of these is LCAT, that we'll talk about it later in the HDL pathway. Very briefly, the lipoprotein metabolism is an overview of lipoprotein metabolism is shown in this slide. So with the different arrows and colors, we discriminate between the exogenous pathway in red, the endogenous pathway in green, and the reverse cholesterol transport pathway of HDL in blue. So briefly, the dietary lipids are absorbed by the intestine, and the intestinal cells, they package these lipids to lipoproteins, which are called chylomicrons, as shown here. The chylomicrons now are secreted, and they transfer their lipids to peripheral cells, During this transport, lipases which are bound to the peripheral cells, to the endothelial cells in the periphery, they hydrolyze the triglycerides to free fatty acids, and the triglyceride content is reduced. And what is left is called remnants, and these remnants return to the liver via ApoE. And the liver now takes all of these lipids after a meal, or if there is no meal, liver makes its own lipids and packages these lipids into lipoprotein particles called VLDL, as shown here. And again, VLDL particles transfer the triglycerides primarily to peripheral tissues, such as the muscle for ATP production, or to the adipose tissue for storage. And via lipases, the triglycerides are reduced in numbers. They are converted to free fatty acids. And what is left is called remnants, VLDL remnants, such as IDL and LDL. So LDL is a particle that is essentially free from triglycerides, contains only cholesterol and cholesterol esters and ApoB, and the LDL particle returns to the liver, and the excess cholesterol is excreted to the intestine via bile acids. And finally, there is the third particle, which is called the reverse cholesterol transport, because this particle now, which involves high-density lipoproteins, HDL, takes back excess cholesterol from the periphery back to the liver. So it does the reverse. So this starts by the secretion of ApoE1 from intestine and the liver. ApoE1 is lipidated by interacting with a transporter called ABC1 in macrophages or in the liver. So the first nascent particles are formed called discoidal, the nascent HDL particles, which are discoidal in shape. Phospholipids also bind to these particles either directly from via ABC1 or by transferring via transfer by PLTP, phospholipid transfer protein, which transfers phospholipids from the VLDL to HDL. The cholesterol that is present in these nascent particles is esterified by LCAT and is converted to spherical, because the cholesterol esters are hydrophobic and they need to be hidden inside the core of the particle. So the particle changes shape in order to protect the hydrophobic cholesterol esters. And this particle keeps collecting free cholesterol and cholesterol esters from the periphery and takes back this cholesterol to the liver via two pathways. One minor pathway is directly via the SRB1 receptor, and the other major pathway is via the CETP enzyme, which transfers cholesterol esters to VLDL first, and then these cholesterol esters are transferred to the liver via the same pathway, the VLDL pathway. So this is an overall metabolism of lipoprotein metabolism, an overview of lipoprotein metabolism. In the remaining of the discussion, we'll focus on two pathways, the VLDL, IDL, and the HDL pathway. That is the good lipoprotein particle, which is HDL, and the bad lipoprotein particle, which is LDL. Continuing on the introduction to lipoproteins, this slide shows the different classes of plasma lipoproteins. Here we see the tissue that they are produced, chylomicrons in the intestine, the VLDL, IDL, LDL, LPA in the liver, and HDL intestine and liver. This actually shows a cartoon. A cartoon shows the difference in their size. We see that these lipoprotein particles differ in size. We have very big particles and very small particles, and in fact there is a correlation between the density and the size. The smaller the density, the higher the size. So chylomicrons are the biggest particles and they have the lower density. The density is shown here. Then we have the VLDL, IDL, very low density, intermediate and low density, and LPA. We have the density is increased as we go to the right. And finally, we have the particles with the high in density but with a smaller size, which are high-density lipoproteins. And these are the more dense because they have more proteins than lipids. Actually, this determines the density, the ratio between lipids and proteins. Here we see the diameters of the different particles. And in general, we now know that high levels of LDL, high LPA, or low levels of HDL are risk factors for coronary artery disease. The lipoprotein particles can be separated by various methods. One is the density gradient ultracentrifugation or flotation. So you take serum and you put it on top of a gradient of a salt such as potassium bromide, and you ultracentrifuge, and every lipoprotein class moves toward the bottom and stops to the density that corresponds to its own density. So if you do this flotation and you start taking samples from the top to the bottom, first you will take allomicrons, then you will take VLDL, then IDL, then LDL, and then finally you take HDL. Also, we can separate lipoproteins by its size exclusion chromatography, such as FPLC, and here we can see a typical FPLC profile of a healthy, a normal human subject. So we see that the cholesterol, the lipid levels, the cholesterol in a healthy human is primarily in the LDL fraction, in VLDL and LDL fraction, and less cholesterol is present in the SDL fraction. This is a typical healthy lipoprotein profile. This is not the same actually in animals which are used for atherosclerosis research, such as mice, which have most of their cholesterol in HDL and less cholesterol in VLDL and IDL. And this is one of the main problems using mice for atherosclerosis, because they have this profile with high HDL, mice develop, it's very difficult to develop atherosclerosis unless you mutate one of the genes of the lipoprotein pathway, and in this case, ApoE. So if you mutagenize ApoE, ApoE is the protein that is important for the clearance of remnants of lipoproteins. We see that if you mutate ApoE, then this profile is very different than this. Most of the cholesterol accumulates in the VLDL, LDL region and less in HDL, and these mice develop spontaneous atherosclerosis. Again, this slide shows the lipids, the cholesterol distribution between species in different lipoproteins. So with red, we show VLDL, with orange LDL, and with yellow HDL. This is the human profile, very high levels of LDL and fewer levels of HDL, but we can see how different it is in different animals. So we'll now focus to one of these lipoprotein pathways, which is the LDL pathway. So the LDL pathway, this slide is an overview of the LDL pathway and some of the genes which play important roles in this pathway. And with numbers in parentheses, we indicate these genes that mutations in these genes, they are causing LDL syndromes, which are characterized by premature atherosclerosis or liver disease. Which are these genes? So the LDL pathway starts in the liver. In the liver, an enzyme called MTP packages lipids and apolipoproteins such as ApoB and other apolipoproteins to VLDL particles, and these particles are secreted in the circulation. The VLDL particles are hydrolyzed, the triglycerides are hydrolyzed by lipases such as lipoprotein lipase, and they are converted to intermediate density, and then by additional lipases such as hepatic lipase, they are converted to the LDL particles. The LDL particles, as we already said, contains essentially cholesterol and cholesterol esters and one molecule of ApoB100. ApoB100 is very important because it is the ligand for the LDL receptor, which is shown here. So this binding between ApoB and the LDL receptor clears the LDL particle, and all the cholesterol goes back to the liver. So we have several genes here. Now, if there is a problem in this pathway, for instance, a mutation in the LDL receptor or in ApoB100, or a mutation in another protein that we'll talk about, PCSK9, in a few minutes, then this particle accumulates in the circulation, it enters the intima and is oxidized, as we already discussed. It is converted to oxidized LDL, which is taken up by the macrophages, recognize it as a foreign substance, and they internalize it. They take it in the macrophages, and accumulation of cholesterol in these macrophages leads to the formation of foam cells and the beginning of the development of the atherosclerotic plaque. Now, mutations in these genes 1 to 5, they cause LDL syndromes, and in the next slide, we show some of the syndromes. Of course, the most well-known is familial hypercholesterolemia, or FH, which is caused by mutations in the gene for the LDL receptor or other proteins such as ApoB or PCSK9. And we also have rare syndromes such as beta lipoproteinemia due to mutations in microsomal triglyceride transfer protein that is required for assembly of VLDL. This is causing a liver disease, accumulation of lipids in the liver. Familial hypertriglyceridemia due to mutations in the lipoprotein lipase, so the triglycerides cannot be hydrolyzed and accumulate, and hepatic lipase deficiencies due to mutation in hepatic lipase. FH, of course, is the most well-known. It's one of the most severe single gene disorders. We see here in this slide that homozygotes for FH, they have very high levels of plasma cholesterol, up to 800 to 1,000 milligrams per deciliter. The heterozygotes, in this case, their parents, they have intermediate levels, and the normal healthy people, they have lower levels of cholesterol. And this cholesterol accumulates in certain areas of the body, creating xanthomas, as we see in this slide. So these two gentlemen, I'm sure we all know them, Goldstein and Brown from the University of Texas at Dallas Medical Center. They discovered the pathway by which LDL is taken up by the cells. Essentially, they discovered the LDL receptor, which is shown here, and they showed that mutations in this protein here can cause FH. That was done in the 70s and early 80s. And in 1985, these two investigators, which are collaborators for life, they received the Nobel Prize for their discovery of the LDL receptor. Now, of course, the discovery of the LDL receptor led to the discovery of the mechanism of statins. Of course, Goldstein and Brown did not discover statins. Statins had been discovered previously, but Goldstein and Brown showed that statins can be used to lower LDL cholesterol in patients with FH. And what statins do, statins are drugs which are competitive but very strong inhibitors of an enzyme which is a key regulatory enzyme of cholesterol biosynthesis pathway. This is called HMG-CoA reductase that converts HMG-CoA to mevalonate. So statins block this enzyme and they block the de novo cholesterol biosynthesis. Thus, they reduce the endogenous cholesterol levels, and as a reaction, the cells activate a transcription factor which is called SRABP, and SRABP goes to the gene, the LDL receptor gene, and activates its transcription. So we have an increase of the LDL receptors in the plasma membrane and a reduction in the circulating LDL particles. So this is the mechanism of action of statins, very, very briefly. And another drug which is more recent and is now used in the clinic is called bempedoic acid. Bempedoic acid actually, it does the same. It acts on cholesterol biosynthesis, but at a different stage, not at the stage of HMG-CoA reductase, but at the stage of ATP citrate lyase, which is an enzyme that lyses citrate to acetyl-CoA and oxaloacetate, and acetyl-CoA is used for the production of cholesterol. So bempedoic acid, which is given to the patient as a prodrug which is called bempedoyl-CoA, is converted to bempedoic. No, actually, the bempedoic acid is converted via an enzyme in the cell to bempedoyl-CoA, which is the active drug. I'm sorry. And this, the interesting thing here is that this drug acts only in the liver, because this enzyme that converts the inactive form of the drug to the active form of the drug is expressed only in the liver. It's not expressed, for instance, in skeletal muscle. And this is the main difference between statins and bempedoic acid. Statins can act also to the muscle, and that has shown to cause some side effects in the muscle, but bempedoic acid is specific for the liver, which actually makes it a more specific drug. Many clinical studies now underway with bempedoic acid, and actually bempedoic acid, I think, is already prescribed to patients. Another very important protein that was discovered relatively recently, not very recently, but relatively recently, is PCSK9. So PCSK9 is a serine protease that was known for some time, but nobody knows what it does to lipoproteins. It was shown to bind to the LDL receptor, shown here, and inhibit its function. How does it do that? It is produced, let's say, in the liver. It is secreted. It binds to the LDL receptor from the outside of the cell and makes a trimeric complex between LDL receptor and PCSK9. So this complex enters the cell via clathrin-mediated endocytosis, and instead of the LDL receptor separating from the LDL and getting recycled back to the plasma membrane, PCSK9 binds very tightly to the LDL receptor and does not allow this separation. So the entire complex, PCSK9, LDL receptor, and LDL, they go to the lysosomes and they are degraded. So PCSK9, when it is expressed in a tissue, always reduces the number of the LDL receptors on the plasma membrane. So, of course, this mechanism was discovered, and also it was shown that patients with mutations in PCSK9 that reduce its activity, they are characterized by very low levels of LDL. And these subjects, which are very rare, very few, they do not have any risk for developing atherosclerosis. On the other hand, mutations in PCSK9, which activate its function, they lead to increased levels of LDL and the fates. So we have these two categories of mutations. First, let's take a quick look at the structure of PCSK9 that contains these domains: pro-domain, the catalytic domain, and the V domain. The catalytic domain actually does not play any role in its function because it is inhibited. The enzyme actually inhibits itself. After a self-cleavage, the pro-domain binds to the catalytic domain and it blocks it. So we don't have any catalytic activity of the mature PCSK9. However, PCSK9, what it does, it binds to the LDL receptor and specifically to the epidermal growth factor homology repeat A, as shown here. The binding is low at the neutral pH, let's say in the blood, but the affinity increases at the acidic pH of the endosomes. So it binds very tightly and does not allow the LDL receptor to get away and get recycled back to the plasma membrane. And these are the two different types of mutations in PCSK9, the gain of function mutations and the loss of function mutations. The gain of functions are rare, 2% of FH cases. They are characterized by high levels of LDL, increased binding to the LDL receptor, and they cause FH. And the loss of function mutations are also rare. They cause reduction in plasma LDL levels, and most importantly, very high reduction in the risk for coronary artery disease. Essentially, the cholesterol levels in these individuals are very, very low. And this slide shows the two different classes of mutations, the loss of function mutations, and this slide shows actually what these mutations do, and these are the gain of function mutations. All these different that gain of function, and this from this side are the loss of function mutations. And of course, after it was discovered that PCSK9 destroys, inactivates the LDL receptor, there was a huge effort by pharmaceutical companies and labs to find inhibitors of PCSK9. And the first class of inhibitors that were developed were antibodies. So the monoclonal antibodies, they bind to the secreted form of PCSK9, and they do not allow the binding to the LDL receptor. So if you use a monoclonal antibody to a patient with very high levels of LDL, the LDL levels will be decreased because the number of the LDL receptors will increase. And it was shown actually that PCSK9 inhibitors significantly reduce plasma LDL levels 40 to 60% in combination therapy with statins. And we'll show in a minute why this combination is important. Another recent drug that also inhibits PCSK9, but with a different mechanism, is inclisiran. This is an siRNA that inhibits the expression of PCSK9. This siRNA, the important thing about this siRNA is that it is liver specific. So it inhibits the production of PCSK9 in the liver because the liver is the main site of PCSK9 production. So it will not inhibit the PCSK9 in other tissues. And it will go to the liver because it is conjugated with a GalNAc, an acetylgalactosamine moiety, which directs this siRNA to hepatocytes that express the acylglycoprotein receptor, as it's shown here. So this complex, the conjugated complex, will enter the cell, will go to the endosomes. The receptor will be recycled back to the cell membrane. The inclisiran is slowly released from the endosome into the cell cytoplasm, resulting in a sustained therapeutic effect. As you can read here, unique dosing regimen of inclisiran, which is initially at 3 months and then every 6 months, which is amazing. You give the drug for 6 months and then you give the drug again after 6 months. Then the inclisiran enters the RNA-inducing silencing complex. We all know how the siRNAs work. Essentially, this is the mechanism of a siRNA function. And finally, inclisiran antisense strand directs risk to bind PCSK9 mRNA strands, triggering catalytic cleavage and inducing production of the PCSK9 protein. So they are novel, small interfering RNAs that lower circulating LDL by more than 50% when added to statins. So this is another very important development in the field. So now, why is it important to give combination therapy, statins and PCSK9 inhibitors? Because we know that at high doses, statins induce the transcription of PCSK9 gene in the liver. So the mechanism is statin inhibit HMG-CoA reductase. They inhibit de novo cholesterol biosynthesis. This leads to the activation of the SRBP2, as we already discussed. And now SRBP2, in addition to activating the LDL receptor, it activates also the PCSK9, because the PCSK9 also have the promoter has binding sites for SRBP2. So you have the activation of both the receptor and the protein that degrades the receptor. So this is something that you do not want. So if you give statins and PCSK9 inhibitors, so you get rid of this branch and you have only this branch, which actually leads to the activation of the LDL receptor without the interference by the PCSK9. And finally, in the remaining 10 minutes or so, I will talk about HDL and the HDL and coronary artery disease. Numerous epidemiological and clinical studies have shown that the levels of HDL cholesterol are a strong and independent risk factor for coronary artery disease. And this study suggested that increasing HDL cholesterol pharmacologically or by lifestyle changes will reduce morbidity and mortality due to CAT, which accounts for 50%, as we said, of deaths of the population. So this, again, is from a large epidemiological study, the Framingham study, showing that the risk for coronary artery disease, which is shown in this axis by numbers 1, 2, 3, 4, is reduced by half, as shown here, for every 20 milligram per deciliter increase in HDL cholesterol. Okay, so this is very clear, and that also led to the formation of the HDL hypothesis, which actually was a driving force for many pharmaceutical companies to develop drugs that increase HDL cholesterol. However, other studies, epidemiological studies, such as the Copenhagen General Population Study, showed that not only the low levels of HDL, but also the very high levels of HDL also increase the risk for coronary artery disease. As shown here, the correlation between the hazard ratio and the HDL cholesterol is U-shaped. So we have increased risk both at low levels and also at very high levels. And the HDL cholesterol associated with lowest mortality, somewhere here, it's approximately 60 to 70 milligrams per deciliter, both in men and in women. So all this data created a confusion in the fields. HDL became very controversial. Initially, everybody thought that HDL is good, so we have to increase it. But then this data came out, and also the data from the clinical trials came out, which showed that all the HDL-raising drugs failed. We'll talk about this in a minute. HDL is very difficult molecule to work with because it's very complex. And actually, this slide shows the entire complexity of these molecules. First of all, it's not one molecule. It's a huge population of particles of different sizes and composition. So we can find in these particles different apolipoproteins, such as ApoE1 alone, or ApoE1 plus ApoE2, or some proteins, some particles have ApoC3, others have ApoE, which are high abundant or low abundant, such as the enzyme cell cut CETP PON1. Different particles have different lipids, high abundant or low abundant, such as phospholipids, cholesterol esters, triglycerides, etc. S1P is one of the lipids which is biologically very relevant for atherosclerosis, but is low abundant. Now, low HDL cholesterol is associated with risk of all-cause mortality, risk for cardiovascular disease, risk of infectious diseases, increase of mortality from sepsis, risk for autoimmune diseases, risk of cancer, risk of diabetes, and low HDL is associated with less kidney function. The major mechanism of HDL is cholesterol transport. I will show you in a minute this pathway, the reverse cholesterol transport pathway, the communication with cells and inactivation of biohazards. HDL can be dysfunctional, and that is the case in certain pathological conditions, such as chronic inflammatory disease or atherosclerosis. And as HDL dysfunction, we divide decreased cholesterol efflux, decreased endothelium protection, increased LDL oxidation, increased serum amyloid A or oxidized phospholipids, or increased inflammatory activity. And as we already said, extremely high levels of HDL are associated with risk of mortality. This is the pathway of HDL reverse cholesterol transport. We saw that very briefly in the overview of lipoprotein metabolism. It was the pathway with the blue arrows. So the liver secretes ApoE1 as lipid-free, ApoE1 and the intestine. ApoE1 gets lipidated by interacting with transporters such as ABCA1 and forms the nascent, the premature particles, which are discoidal in shape and consist essentially of ApoE1 and some phospholipids. This particle gets cholesterol from the periphery, such as the macrophages or other tissues, and is becoming bigger and is becoming spherical because of the action of LCAT, which, as we said, esterifies cholesterol and makes the particle spherical. The mature HDL particle now, they transfer the excess cholesterol back to the liver via two pathways, one directly via SRB1 and the other indirectly via the VLDL-LDL pathway. And here the major role plays the enzyme called CETP. And why this is important? Because CETP was one of the major targets of the pharmaceutical companies to increase HDL levels, because from this slide we can realize that if you inhibit CETP, then you can prevent actually the transfer of cholesterol esters back to the liver, so we have accumulation of HDL in the circulation. Now, this proved not to be good, but that was the idea behind the development of the CETP inhibitors. And mutations in the genes shown in asterisk, such as ApoE1, ABCA1, and LCAT, cause very low HDL syndromes and premature atherosclerosis. In the case of APOE1, we have hypoalpha lipoproteinemia and amyloidosis, but this is something different. In the case of ABCA1, we have Tangier's disease, and in the case of LCAT, we have familial LCAT deficiency and fish-eye disease. And these are two slides just showing some of the clinical features of ABCA1 and of LCAT, but I will not stay in this because I would also want to emphasize that although mutations in these genes have been shown to cause to be associated with premature atherosclerosis in some of the cases, not all, overall, the GWAS study, the genome-wide association studies, failed to show a correlation between polymorphism and coronary artery disease, as shown in this slide. So it was different. So from the genetic point of view, it could not be established that HDL is causal to atherosclerosis, that mutations in the HDL genes such as CETP or ABCA1 or LCAT in the general population, they cause coronary artery disease. That could not be verified in genome-wide association studies. And in addition to that, all clinical trials with drugs that significantly raised HDL cholesterol levels, such as CETP inhibitors or niacin, or nicotinic acid, another molecule that increases HDL, have failed to show clinical benefit on top of statins. So you see the p-values here for these alleles, different alleles. So you see that although the p-values for the HDL cholesterol are very, very low, so these polymorphisms are associated with low levels of HDL cholesterol, there was no association with coronary artery disease. But we know that from various studies in animal models and in cell models that HDL particles have various atheroprotective functions, such as macrophage cholesterol efflux, antioxidant functions, anti-inflammatory, anti-apoptotic, NO production effects, wound healing, antimicrobial and antiviral, antithrombotic, inactivation of T cells, and protection of beta cells. So all these atheroprotective functions were shown to be a characteristic of the HDL particles. Of course, one lack in knowledge, one gap in knowledge here is that we don't understand which type of particle is responsible for all these different functions. And I will finish, and I believe this is my last slide. Although the clinical trials failed to show that there is a benefit by increasing HDL cholesterol, the HDL field actually is still alive and active, and now the focus is on the development of new drugs which are based on HDL functionality instead of increasing the HDL concentration. And some recent developments in this field is the development of drugs based on APOE1, or these are essentially reconstituted HDL particles containing pure APOE1 and phospholipids. And there are two types. One is containing wild-type APOE1, the SER001, and the other containing the APOE1 Milano plus phospholipids, MDCO-216. These were used in some clinical trials with some promising results. And very, very recently, it was shown that also recombinant LCAT can be given to patients with the syndromes of LCAT deficiency that are characterized by low levels of HDL. And I will sum up everything in 2 slides. So coronary artery disease is the leading cause of deaths worldwide. Coronary artery disease is caused by atherosclerosis, that is the gradual thickening of the vessel lumen due to disposition of excess cholesterol. Dyslipidemia, due to imbalance in lipoprotein metabolism, is one of the major risk factors for atherosclerosis and coronary artery disease. The study of rare monogenic syndromes of dyslipidemia had led to the discovery of new genes that control LDL levels. And here we talk about rare syndromes where we have mutations in PCSK9, which are very, very rare. Inhibitors of PCSK9, monoclonal antibodies, and siRNAs constitute the most promising class of drugs for the prevention and the treatment of coronary artery disease in combination with statins or bempedoic acid. Low HDL cholesterol is an independent risk factor for coronary artery disease, but very high levels of HDL also predispose to coronary artery disease. Many proteins participate in the metabolic pathway of HDL, including apolipoproteins, plasma enzymes, and membrane receptors and transporters. Mutations in the three key proteins of the HDL pathway, ApoE1, APC1, and LCAT, account for a small fraction of low HDL syndromes. And GWAS studies fail to associate HDL genes with coronary artery disease. And furthermore, clinical trials with HDL-raising drugs also failed. Clinical trials with molecules that increase HDL functionality are promising. And finally, understanding the proteins, the genes, and the lipid biomarkers that affect HDL will facilitate the development of novel HDL-based therapies. So I will finish here. I would like to thank you very much for your patience and for your attention. And I could take questions. So I think, Miron, we can leave the presentation open, or should I close it?
Yeah, no problem.
Thank
you so much.
Okay.
Thank you so much for all these novel details on more traditional information, and then everything new that has been discovered in previous, I would say, decade. I would also like to welcome Paolo and Yael as panelists for this webinar series. We can open a question. Maybe it's going to be easier if we start with questions, and then we can move forward to questions that are asked by people from the audience.
Is there anything in the chat, or?
Yeah, I will go through, but currently I don't see any direct questions anywhere. So, Paolo, Yael, if you have any questions to start with, or maybe I can start, doesn't matter.
You can start.
Okay, thank you. Well, Dimitris, you mentioned that LDL and HDL are currently, not currently, but they used to be the biggest two focuses of this story when we talk about lipoproteins and lipoprotein metabolism. And we know that in popular language, we have like a good cholesterol, which is HDL, and a bad cholesterol, which is LDL. And of course, from the more professional side, we know that we cannot be as black as white, talking about just the good and just the bad, because there is context behind that we need to consider. But regarding the progress that was done in the previous years, we can see that in the latest guideline by EAS regarding atherosclerotic cardiovascular disease, it is not just LDL that is targeted anymore. Now we have this complex term that is called non-HDL. So if you just can emphasize a little bit and explain what is non-HDL and why we need to go a little bit above and beyond LDL as a traditional risk marker, and discuss about non-HDL.
Yeah, essentially, non-HDL is everything that contains ApoB, as a matter of fact. If you remove HDL, what is left is all the other particles, which are VLDL, IDL, LDL, LPA. So of course, right now the focus goes on LDL, but now we know that other lipoproteins can also be increase the risk. So it's better to define the risk factor as non-HDL instead of LDL, because other lipoproteins are equally bad. So we cannot say that only LDL is bad. So for instance, we know that from our animals in ApoE knockout mice, we have huge levels of VLDL and very low levels of LDL, and these mice have tremendous atherosclerosis, spontaneous atherosclerosis, with VLDL only. Now, since you mentioned good and bad cholesterol, some people tend to refer to HDL as the good cholesterol. This is totally wrong. In HDL, cholesterol is not the good component. Okay? In LDL, cholesterol is the bad component, but in HDL, cholesterol is not the good component. The good component in HDL is the protein components, not the lipid components. S1P also, but it's very minor. lipid, sphingolipid in HDL, but all the good things that HDL does is through ApoE1 and primarily. Okay, so we should not refer to HDL as the good cholesterol. The good lipoprotein particle is the correct term, but the good cholesterol is not correct term. Okay, so this is something that we have to keep in mind. For LDL, yes, cholesterol is bad, but for HDL, cholesterol, we cannot say that it is good.
Yes.
But again, yeah.
Okay. Let me just check if there are any other questions. Paolo, would you like to?
Yes. Thank you, Dimitris, because really this was a very illuminating overview of all the biochemistry and the applications of the biochemistry of lipoprotein. So very, very interesting, very complete. So clearly, when you see that, well, you can measure LDL, but then you can have oxidized LDL, small dense LDL, I mean the size, and then you can measure ApoB. So, you start thinking about a large set of descriptors of the situation.
Yes.
So clearly, we cannot do everything. We need to have a balance because, of course, now we are all together linked in research projects, and in research you can do, I don't say whatever you want, but you can do, let's say, more sophisticated approaches and so on. But in practice, maybe is it just useful, as Miran said, to approach a non-HDL without measuring ApoB, or you see advantages in trying to, let's say, describe the size of the and density of the LDL, or oxidation, or ApoB?
These are very important also, but these are still not in the clinical practice, and I don't know how easy it is to measure all these different particles in the clinic. So the easiest thing is to measure LDL cholesterol, or perhaps non-HDL cholesterol, but it would be nice if you could measure the really bad particles, such as the oxidized LDL and everything. But I don't know if it is right now. It's in the clinical practice. I think it's not. It's not. So ideally, it should be included in the clinical practice. And now, since we are talking about these different LDL particles, we can do the same for HDL particles, because we know that HDL particles are also very heterogeneous, and some are better than others. So usually what we do is we measure HDL, but this number, let's say 50 milligram per deciliter, I don't know what it means and how the GP can use it. But if you measure functionality, such as cholesterol efflux capacity or PON1 or antioxidant activity, then it will be much more useful than measuring HDL concentration. But still, again, this is not in the clinical practice. So many things need to be changed, and many new analyses that focus more on specific particles need to be included. I don't know how much it's going to take, but this is a must, I believe.
Yeah, so maybe it's a matter of having different levels of clinical approaches. So general approach by the GP, and then in specialized or in very higher risk patients, maybe.
Yes, you can send the samples to specialized labs to measure these particles. Of course,
very higher risk patients, in order to have a better outcome. Thank you.
Yes, exactly, absolutely.
I would also just like to draw attention of the colleagues that are watching the webinar on your slides, where we can see, or on your slide where HDL has actually reverse or U-shape epidemiological curve, which means that we should not be, we should be aware that also high levels of HDL are not good. So with the HDL, the story is even more complicated than with LDL, because so far we don't have any information that extremely low concentrations of LDL have any unbeneficial effect or adverse effects in humans. But with HDL,
we have some information from patients with PCSK9 loss of function mutations, because these very few individuals have LDL cholesterols which are in the range of 5 to 10 milligram per deciliter, and they do not have any symptoms. These individuals, so who can go very low
with LDL.
in LDL.
Yeah.
But we cannot go very low or very high in HDL.
Yes. I think that is very important take-home message, because sometimes people really, I mean, you can see here that the shape of the curve is changing around 1.9 millimoles per liter, or in the other, depending on males or females, 2.4 millimoles per liter. And these are not huge, very huge numbers when it comes to cholesterol concentrations, because previously, I remember when I was during my undergraduate studies, these numbers where HDL was considered to be bad was like 5 millimoles per liter or 4 millimoles per liter. But now we see that these numbers are actually quite lower. And personally, me, from my surroundings, I know some people that have concentrations like 2.2 millimoles per liter for HDL. And then also, when you calculate the risk, sometimes there are these indexes that are frequently used in clinics, like HDL over LDL ratio. And then this ratio turns out to be very good if your HDL is too high, but it's quite misleading, because extremely high values of HDL, but not just extremely high values of HDL are also not that good. So I think this is a really important message for the future generations of our listeners here, that to be aware of that, that we should not go over the limit with the HDL also on the upper part of the curve, because it also brings risk. And it's very nice to see these graphs are published in this year European Heart Journal issues.
Yes, and also you have increase in other diseases, such as dementia and other, not only cardiovascular disease. If you have very high levels of HDL, you have increased risk for other diseases as well.
Yeah.
So,
do we have any other questions regarding this fascinating story between lipids? Let me just.
I have a very short question, but it's really just to satisfy my curiosity. I was intrigued by the slide you showed, Dimitris, of animals and their cholesterol levels. And all we learn is that mice don't get cardiovascular disease unless we genetically modify them. So now I'm thinking, are there animals that do get cardiovascular disease other than humans?
Yes, of course, there are animals. So the closest the lipoprotein profile to humans, the better the models are. For instance, the pigs, which are shown here, have a profile which is very close to humans. And these pigs are very good for atherosclerosis studies. They develop coronary artery plaques and myocardial infarction. Mice are very different from mice. However, if you mutagenize ApoE, or let's say the LDL receptor, then the profile is very atherogenic, as shown on the right-hand side. That's why I put it here on the right-hand side. Of course, this will be the subject of another webinar when we will talk about models of atherosclerosis. So this will be another webinar, not the immediately after that, but one in the future of the webinar will be talk about animal models. So on the right,
that would be extremely interesting.
Yes, you see that the LDL receptor. So this is a condition that mimics a face, essentially. So you see the profile here is very close to humans.
Yeah.
And these mice develop atherosclerosis, but on a high-fat diet. Okay, and ApoE also that have this profile, they develop much more atherosclerosis than the LDL receptor knockout mice. So these ApoE knockout mice have very high levels of VLDL, okay, instead of LDL. They have LDL, but VLDL as well. And these are different models. Now, why mice do not develop atherosclerosis is because they lack CETP. They don't have CETP. So they have huge levels of HDL, as it's shown here. What is it here? The yellow. You see how big the yellow bar is? Because they don't have CETP. This enzyme that everybody tried to inhibit a decade ago, and they did it, but these inhibitors failed for various reasons. Probably The main reason is that the HDL particles, they were dysfunctional. They could not work. Okay, so if you block the catabolism of HDL, this is not the way to go. The way to go is probably to increase the rate of reverse cholesterol transport by activating ApoA1 or APCO1 or LCAT, but not by inhibiting the catabolism. This is not good for the organism.
Thank you so much.
Sure.
Yes, Dimitris, thank you so much. Thank you for also announcing that in the next 2 webinars, basically, we will also covering a more of these models that are related to that are good for investigating cardiovascular disease in cell lines and in animal models. So Paolo will give a lecture about the relevant cell lines that are used for ACVD on, I think, 24th of October. And then after that, we will schedule another one where you can cover these various animals models. And with that, we will probably finish up with this part where we dive into the stories behind the atherosclerosis, and then we will move on a little bit more about various omics approaches, learning about new technologies and what is out there currently that can be used for various omics analysis. So unfortunately today we didn't have any questions from the audience, but the webinar will be also available online until next week, probably, because WebEx needs some time to process everything and to sort things out. So if anybody, of course, has any questions for the future, just drop us an email and then we can communicate through that. But I think this is most important for younger listeners currently, just to be engaged as much as they can. So even if the subject is overwhelming currently, don't be afraid to go again through the lectures. And then if you find anything that is not, that you are not understanding well, please contact the lecturer. And I think that you can continue more in more private, I would say, environment, the communication, because all these lectures are for you and just to clear things up, at least those things that are currently not very clear for you. So with that, I think
Dimitris, if I can say a word, just to reinforce the concept that these are not one-way webinars. These are just getting together, clearly online. So I really emphasize the fact that, as you said, it's very important that younger researchers will participate, will comment, share their experiences in these webinars, because we are not just, this is not just, seniors are talking to juniors. This is a dialogue, okay? So please come and see and ask.
Yes.
Exactly.
Thank you so much. I wish everyone a nice afternoon, nice weekend. See you in a couple of weeks. You will get also the invitation. So see you soon. Bye-bye.
Bye-bye. Thank you.
Bye-bye. Thank you.
Bye-bye.